Thank you for coming. My name is Lisa Galea, and it is my distinct pleasure to introduce Dr. Anne-Marie de Lange, who I didn't ask how to pronounce that properly, but she'll tell me later. I'll have a quick introduction, and then we'll get right to Dr. Lange's presentation. First, I'd like to acknowledge that we live, work, and play and participate in a community on the unceded, ceded, and traditional territories of the 203 First Nations, along with the 38 Métis chartered communities, each of which possess their own unique traditions and history here on this land of which I dwell, that we refer to as British Columbia. We acknowledge the importance of the Truth and Reconciliation Commission of Canada's call to action, the United Nations Declaration on the Rights of Indigenous Peoples, and the BC Declaration on the Rights of Indigenous Peoples Act. In all of our work, we're committed to ensuring Indigenous women's rights to health and safety and equal opportunity to participate in a manner that recognizes and respects Indigenous cultures and traditions. And I recognize that I am from Canada, but many of you are not, and I just urge you to look and learn about the Indigenous peoples on lands of which you dwell. So I'm actually joining from North Vancouver, which is part of the unceded, traditional, and ancestral homelands of the Coast Salish peoples, the Musqueam, Squamish, and Tsleil-Waututh First Nations. And I lead the Women's Health Research Cluster, but there's lots of people supporting the cluster, which is fantastic. We have many members worldwide across 17 countries. And we have a number of events, one of the seminar series of which you're part of right now. We also have a Women's Brain Health Conference that's starting up again next month. It's a virtual conference. It's just every 2 weeks, I think 1 or 2 hours. And it's on hormonal contraceptives and effects on the brain. So if you're interested, please take a look at that. There's lots of awards for trainees too, if they'd like to attend that. And just take a look at our website. We have a lot of different outreaches, like blogs. You can get involved that way. And we have a podcast as well. And we're a multidisciplinary group, and we invite everyone. We're trying to be very inclusive, so please do become a member and donate if you so desire. So with that, I'm going to stop sharing my screen so that you can hear from who you really want to hear from, which is Dr. Anne-Marie de Lange. And so I'm going to read her bio out and her fun fact. Dr. de Lange has a background as a clinical psychologist with a PhD in neuroimaging and brain plasticity from the Center for Lifespan Changes in Brain and Cognition at the University of Oslo. Always wanted to go to Norway and see those fjords. She studied brain aging and large-scale population cohorts as a postdoctoral fellow at the University of Oxford. And in 2021, she established the research group FemiLab at Lausanne University Hospital in Switzerland. FemiLab focuses on how factors such as reproductive history, endocrine aging relate to brain aging trajectories and disease risk, and how these factors interact with genotypes and common genetic variation, and how these associations may contribute to epidemiological differences in risk and disease. And actually, I'm quite Anne-Marie, you can share your screen anytime. I'm quite a fan. I'm a little bit tongue-tied at the moment because, you know, it's like meeting your idol, even though she's many years my junior. And she, her fun fact, because she's amazing and has done so much, and she has some fantastic publications, which I really urge you to read. Her fun fact, she has a couple of them. I'll give you the, she wants to combine a women's health conference with a late night karaoke session so that we can sing a duet together. But then I told her I'm not actually a very good singer, but I would totally love to do karaoke with her. And apparently she has a family of foxes in her backyard that she is learning how to treat because they have some diseases. So I mean, she is very talented, and I'm very proud and very happy to welcome her to the Women's Health Seminar Series. So take it away, Anne-Marie, and we'll leave questions for the end, so please put it in the chat or put up your hand at the end. And I will leave my camera on, and I'll be looking at my phone, but it's only because I'll tweet out things, only good things.
Thank you so much, Lisa. That was a wonderful presentation and a very good introduction. Just to ask, can you now see my slides and nothing else? So you don't see all the mess behind the slides on my screen.
I do not see any mess in your house or on your slides.
That's great. Excellent. Okay, so good morning to everyone and good evening to me. because I am indeed in Switzerland and it's 9 in the night. But I'm delighted to be here, and I'm very excited about this little talk. And I hope it will be fairly useful for you as well. So I will talk about a big data approach to women's health research. And what I plan to do is to try to shed some light on a few questions. The first will be, why is it relevant to talk about big data? What is the UK Biobank and how can we use it? And then at the end, I will say a little bit about how I look at the future in terms of using big data for women's health research and how we can move forward, basically. So let's just start with some very general aspects about big data. These data sets are very often based on population cohorts. And basically what that means is that in comparison to very targeted data collection, where you collect data because you want to look at a specific disease or something, population-based data is often a bit more general. However, they often start with some idea of wanting to explore some disease in the population, but they often collect so much data that it really expands and people start using it for lots of different purposes. So in some ways, you can say it's quite general, but there's often an initial idea behind them. And sometimes participants would be recruited based on where they live or which year or even which week they are born in. So this is great for us, obviously, because we like to see data that is, representative of the population. We like to see big data that can cover lots of different aspects, but we must also admit that these data sets are very often not very representative of the entire population. And we know, for example, that people who participate in research in general are often more healthy. They are often they have higher education. And there are also other issues, such as, for example, with the UK Biobank, where I think 97% of the participants are white. So these are not particularly generalizable at all, actually, but they still give us a pretty good overview of the things that we like to have a look at in terms of research. And one thing that is really nice, I think, is that many of these data sets are made available to researchers globally. So you can apply for data access and you can download the data and start using it, even though you didn't participate in collecting it. And this is extremely important in terms of replicating your findings across data sets and cohorts. However, it also requires quite a serious lab infrastructure if you want to do it properly, because you can only imagine downloading loads and loads of raw MRI data and then trying to harmonize all the MRI processing streams with maybe a smaller data set that you collected yourself, all of which sounds great, of course, but it requires a proper data management team. So it's not so easy for everyone, right? This is something that you can do if you really have the infrastructure. But anyway, with these huge data sets comes statistical power, which we absolutely love, of course. But it's also extremely easy to find significant results. So what we often see is that we have these significant p-values all over the place, but the effects are often very, very small. So this is a bit mind-boggling sometimes, because we have to ask questions such as, what does this even mean? If you look at these normal scatter plots, when you use the UK Biobank data, you often see that you have very, very large variation. And then if you plot your effect like just a fit line, it will basically look flat. You will not even be able to see that there is a positive association if that's what you saw, because it's so tiny. It's still there, right? And it might be important, but it's also often very small. So we have to ask different questions when we have these big data sets, because it's so easy to pick up very subtle effects. And that's due to the statistical power, basically. Another quite nice fact about these big data sets is that they often have a very wide range of health data, a very wide range. So in the UK Biobank, for example, massive amounts of data on all sorts of things. And this is great because it enables us to do, we can model lots of different factors, behavioral and clinical, biological, and we can also use methods to see how they co-vary and maybe, link to brain patterns with aging, things like that. But it can also be fairly overwhelming because sometimes we want to just look at something specifically, right? We have two factors, we want to know how they relate, and then we end up in a situation where you basically could correct for absolutely everything else about the body because you have the data available. And that doesn't really make sense, right? So we have to think a little bit differently in terms of whether we can even ever actually expect any effects to be specific, right? Or independent of everything else when it comes to health. And this was a bit new for me, I must admit, when I started working with these big data sets, that I really had to think differently about health. Okay, so the UK Biobank, I've already mentioned it, and I'm sure that many of you are familiar with it already. But just to say, like, very briefly what it is, and I don't work for them, like I'm not I'm representing the UK Biobank, so I don't know everything. I'm just giving you an overview from the user perspective, basically. So they have genetic and health information from 500,000 UK participants. It's pretty big. And the data collection is still ongoing, it's rolling. So they are following people up over time and new data is added regularly. And you can access this data through their online application. You have to have a project where you say what you're going to do with the data and which variables you would like to use. But other than that, it's fairly straightforward, I would say, to gain access to this data. You need to make sure that you have space to store the data, etc. But it's absolutely accessible, I would say. So this is a big initiative, obviously, and they have more than 200 staff members who work with this across the UK. And they have funding from several bodies like the Wellcome Trust and the Medical Research Council. And the hope is that this will just continue, right? They will just continue to collect data and more data will be made available. This is maybe one of my favorite things actually about the UK Biobank, and that is that you can just go to their website and you can find so much information. Even if you don't have an application, you don't have to be approved as a researcher or have access to the data. You can just go to their website and browse everything they have. And I think it's so nice because it's very transparent, and you can always refer back to all of these overviews online. So you can look for example, specific variables or categories. And here is one example, which I thought was a bit funny because it's sex-specific factors. And if you look at the number of items, it's just a little bit funny, but yeah, it's a good example of how you can find, just look up what they have basically online. And if you go to something specifically like menopause status that I'm showing here, you also get really nice distributions of the responses. You get some information about how the data was coded, how it was collected, whether it was a follow-up to a different questions, all sorts of stuff. So when you sit with your data set, which is often a subsample, right, of the whole data set, then you can very easily just go to the website and refer to the overall status of the data that has come out. So I think this is extremely useful, and I'm very grateful that they have done it this way because it's super transparent, really, really nice. Another thing is that all the approved researchers or all the projects that have been approved are also available online. So you can look at what's already going on, and it could be a nice way to find potential collaborators. keep an eye on what people are doing. And yeah, it's really nice. This is just an example from the lab that I am working with in Oslo. So all of that said, in general, the question is sort of how can we use it, and how can we use it for the research questions that we are interested in. And I think one thing that is quite obviously important is the age range of the participants, because it is midlife and older age. And I think it's something probably 40 to mid 80s or something, if I remember correctly. So this means that we can't really model what's going on with brain development, for example, in adolescence, but we can look at what's happening in midlife and older age. And for us, it's not the worst, right? Because we have the menopause transition more or less covered, or at least to some extent, which is actually quite rare. And they do also collect historical data, so you can get information about health issues earlier in life and such, so it's not entirely restricted to midlife and older age. You can also get historical information on these participants as well. So the MRI subsample so far, I think, is 40-something thousand, possibly a bit more than 42 now, data that has been released. And they also have follow-up data released on a few thousands. I have never used that, actually. I hope it's on the horizon. But they keep, they keep adding these the new batches of data so that we can easily access and look at it. So it's really amazing. There's a good balance between male and female participants, and they have female-specific variables. And some examples would be reproductive history and age at menopause. And I'm mentioning these two factors because these are factors that we have used quite a lot in our research. So I plan to give you a little overview of some of the studies that we have done with using the UK Biobank data across some different topics that are more or less related, but still a bit different from each other. Okay, so when it comes to brain development and aging in particular, we have sort of, I'm saying going back to basics because we're using new methods, or relatively new methods, to model trajectories with age, basically. And one of the things that we've used quite a lot, and I'm sure many of you have heard about, is brain age prediction or age prediction. And this is typically something that we can definitely do with big data, but it's a little bit trickier with smaller data sets. And I'm going to try to very briefly just explain to you what it is with the help of this very nice figure that Claudia made. So basically what we do is that we train a machine learning model to learn how the brain looks across age. So we're telling this algorithm to learn what are the patterns and how do they change with age. And then you can use this model, which is then sort of a buildup of a normative trajectory. Then you can take this model and you can use it to predict age in other samples or unseen data. And basically what you get from it is a measure of how old someone's brain looks compared to what is expected for their chronological age. And in this example that you see on the figure, you see an individual who has a chronological age of 50, but their brain age has been predicted to be 45. And what that means is that their brain age, as we call it, is a little bit younger than what is expected for their age, which is good, right? So then they get this brain age gap, which is what we call it, of -5 years. And I guess what's the most good, or the best thing about it, is that when we have so much data, which is really what we have with the UK Biobank, with loads of participants, loads of MRI data, and here we can just put all of this into this machine learning regression, and then the model will learn like very subtle patterns in the brain that varies across age, across regions, across modalities and tissue types. And it could be even nonlinear and linear patterns all together. And then you end up with one estimate per individual, which is very easy to work with, obviously. And if you compare it to, let's say, other traditional summary measures, such as total gray matter volume, for example, which is also often used, I think the main advantage of the brain age measure is that you can put so much data in, and the machine is actually learning something as well. So you're not just averaging across everything, you're actually doing a little bit more. It might sound a bit complex and difficult, but it actually isn't that hard, because it is just a regression in the end, right? So you're predicting age based on brain. You just need to know how to code. Okay, so yeah, just to go back a bit to the brain age measure, it's quite nice to use it when you look at, for example, patient data, right? So you have this normative trajectory and you look at deviations. So what we very often see is that patients with, for example, dementia will have older brain age compared to what is expected for their chronological age, as we would expect, right? But you can also use this estimate to look at associations with behavioral or other types of factors that we're interested in. So we have used it for different purposes, and one example is reproductive history. So we ran some studies, it's actually a few years ago now, time flies, where we looked at number of previous childbirths in the UK Biobank women and how this related to brain age and older age. And it was quite interesting because we found that a higher number of previous childbirths was linked to a younger brain age and older age, or midlife and older age, I guess I should say, because they're not that old. They are spanning from, I think, around 40 to 80 here as well. So if you look at the plots here, you will see that there is this negative slope, and you have the more childbirths they have, the lower the brain age is. But what you can also see is that for the groups of women who have given birth to more than 4 children, the statistics are not as high. They're still fairly high. Everything is relative. But I think we're talking here a couple of hundred women, whereas for the group of women with 2 children, it would be more like 5,000. So it makes a difference. And you can see then in the error bars as well that we cannot conclude as strongly when it comes to these groups. So even when you have enormous amounts of data, you still end up in situations where actually the data wasn't enough. Right, so when you start looking into groups, you still have that challenge. Anyway, we couldn't explain this finding with any confounders. We tried to basically correct for everything, at least a lot of things, not just lifestyle and health factors, but we also looked at pregnancy complications and stillbirths, all sorts of things. And these results seem to be quite stable, actually, across different types of analysis and corrections. So what it could mean is that obviously this is a cross-sectional study, so we can't say anything for sure, but it could mean that those effects that are seen across pregnancy and in the postpartum period in other studies, that there could be some indication of these effects many, many years after these women have given birth. So one follow-up study that we did as well, just very briefly, we found that these effects were actually most prominent in subcortical regions in the brain, which we thought was even more interesting because there are so many studies showing effects exactly in these overlapping regions. And I'm sure I've forgotten lots of citations here, my apologies. There are so many good studies on this, and yeah, it's just super interesting to see that there is some kind of, convergence across these very different approaches, I think, to studying pregnancies, right? So then we're still asking these questions of what are the mechanisms that underlie these results that we see so many years later, and it could be linked to hormonal and immune changes, but it could also be linked to experience-dependent brain plasticity due to parenting. And I think the field is sort of moving towards that it's very likely a combination. And now we're seeing some nice results from fathers as well as mothers, where there might be some different brain changes, but clearly there are effects across both mothers and fathers. So very interesting stuff going on in this parental brain field, I think. Right, so I will move us to another area of research that we've been quite interested in recently, which is cardiometabolic health. So we're still looking at brain age now because we're very happy with this as an outcome measure. It makes life very easy for us. And when we compare it to other measures of, white matter or gray matter, whatever, it looks very similar often. So I don't think we have a lot of reasons to doubt this as a measure. It's a little bit complex sometimes to understand how it's calculated, but in general, it doesn't show anything completely different from more traditional things. It's very similar. So one study that Sivanya, who's the postdoc in my group, did and published actually this year, she's looked at sex differences in body composition and brain age. And this was very interesting because if you look, I know it's a bit busy, this plot, but I'll do my best to take you through it. If you look at this here column where you see brain age gap versus waist to hip ratio, you will see that for both males and females, you have this positive association. So the higher the levels, the worse the brain age, or the older the brain. And then if you look at BMI and body fat percentage, it doesn't look like that at all. So for females, it's actually quite different. You don't see this expected association between higher BMI and older brain age. So what could that be about? So we're asking the question then whether body fat could be less detrimental for women's brain health compared to men, or what's going on. And for the astute in the audience, you may also have noticed that we had some associations here with the brain age gap and ApoE4 as well. And that's not my focus, but I just wanted to mention that because you see that basically we don't see very much at all here, very, very tiny effects. And I just think it's important to mention that this is a very healthy sample. We have excluded anyone with neurological issues and mild cognitive impairment, dementia, etc. So it's a very healthy sample here. So that could be one of the reasons why we don't see anything for ApoE4 status here. So back to the body composition then. We follow this up in a very recent study by Louisa, who is a PhD student in my group. And she looked at whether different types of adipose tissue post-menopause could potentially have different roles in brain health. It is starting to get quite complicated, but what's really nice in the UK Biobank is that you can also access body MRI scans, actually. So you can look at specific adipose tissue types. So here she looked at visceral adipose tissue versus abdominal subcutaneous tissue. These are different types. You can see that the visceral fat is lying or surrounding the organs, and the subcutaneous fat is just beneath the skin. And there are some indications that the visceral fat can be more dangerous in general. So we were wondering, could there be some differential effects here? Because we also know that after menopause, adipose tissue is actually important for estrogen production. However, though, what Louisa found was that both of these tissue types were linked indeed to older brain age and white matter hyperintensity load. So higher levels here were linked to older looking brains. Another very interesting finding here, though, was that these effects were primarily actually seen in women with longer reproductive spans. Now it's getting even more complicated, but what we know, or at least what we see in the literature, is that cumulative lifespan estrogen exposure can actually have implications for both the body and the brain after menopause. So whatever happened before menopause can still influence how you do later. So in this case, it might mean that greater lifetime estrogen exposure in combination with higher levels of adipose tissue or body fat could potentially represent a risk of adverse brain health in older age or post-menopause, and or less lifetime estrogen exposure. So if you get your menopause earlier, could be linked to some type of beneficial effect of adipose tissue later because of some compensating mechanisms where the estrogen production is actually all right, and it compensates so that you don't see detrimental effects. Super, super complicated. We've struggled a lot with this paper, but it is coming out. And we're looking forward very much to follow it up with, more, hopefully maybe not more detailed, but hopefully more clever studies. For example, it would be really nice to see this modeled across the menopause transition. So what we basically can conclude here is that there are certainly some very complex interactions between cardiometabolic, neural and endocrine processes. So I just want to circle back to the endocrine processes, because one thing that was found across both of these studies that I've just mentioned was that an earlier age at menopause was linked to an older brain age. And this aligns with a lot of research out there where, a longer reproductive span is often shown to be better. More estrogen exposure for longer is also shown to be better. It's not always that straightforward. We have some controversial findings too, where, people see different things, but overall, this was not the very surprising finding, I would say. And then I suppose the one natural question is, what about hormone replacement therapy? Because when women experience menopause and the decreased estrogen, it's quite common now to basically compensate and treat symptoms with hormone replacement therapy. And that is also something that is not completely clear from the literature in terms of what effects this can have on the brain. So in 2020, Claudia and I published a study. I know Lisa doesn't like it. I'm sorry, Lisa. It's very interesting. We looked at basically hormone replacement users versus non-users. So a very simple measurement, basically. And what we saw was that actually users had older brain age compared to non-users. The effects are super tiny, like they're very, very small, but they keep popping up, in the UK Biobank. It's just what we see. And it was quite unexpected, but the more we sort of look into the literature, the clearer it is that nobody fully knows, right, what's going on here. So another thing that also came out of the same paper was that when we looked at estrogen levels in the blood, it was measured a few years before the MRI scan, but it was the best that we had. And then we saw that actually the higher estrogen levels in the blood was linked to older brain age, but only in ApoE4 carriers. So we're just thinking that, if the effects of hormone replacement therapy depends on genetics and all sorts of different factors that we don't always have, accessible in our data sets, and we don't even know always how to model or what to look for, then maybe this can explain why studies show different things. it has been very complicated really to look into this, and we're still not sure what this means. Also because again, the study is cross-sectional, we can't really conclude anything. And we have actually prescription data available in the UK Biobank, but this is super complicated as well, because even if someone was prescribed a certain dosage or type, we don't know if they actually took it, right? So there are all sorts of questions related to that data. So Claudia is still working on this, and, you know, hands up to her, because it's not easy at all, but hopefully something comes out of it, and yeah, we're trying to make sense of it for sure. Okay, so I've just showed you a few examples of some of our studies, and they are, in my opinion, very complicated, and it's not been easy to try and figure things out here and understand how to move this, you know, these analyses forward. So just a shout out to the team who are working extremely hard. I'm very impressed and grateful. I now also see that my picture is on the slide as well, which I hadn't noticed before now. The point is not to thank myself, but to thank the rest of the team. Sorry about that. Yeah. Okay, so I've talked a little bit generally about, you know, big data, the UK Biobank, and some of our studies, how complicated it all is. So I would like to just take a moment to talk about some general aspects when it comes to big data, women's health, and where to go next. So again, back to these population-based cohorts. UK Biobank is unique. I mean, it includes so many female-specific factors that we very rarely see anywhere else. So I think, you know, in that sense, it's an excellent cohort to use if you want to do these types of studies. But there are still some issues, as I was saying, you know, it's not that easy with the hormone replacement data, and it's not that easy with, for example, we don't have any data on breastfeeding, etc., etc. It's not ideal, but it's very, very good still. But this is an exception, right? Usually they actually don't have anything at all. And this is something that we've started to discuss a bit recently, right? How can we actually influence these data collection processes? Because these are massive initiatives. And if we can tell them what we need, you know, we might get through to them, and maybe they can start incorporating some factors and variables that we're interested in and that we think are important for health as well. And this is also related to my second point here, because we all know how expensive and time-consuming data collection can be. So while we wait for, you know, all our grants to be successful, it would be very nice if we could help, you know, with influencing and incorporating relevant variables into these ongoing processes that are absolutely massive. Again, in relation to all of this, so it's hard to get funding full stop, but it's also particularly difficult to get funding for female-specific studies. I think we know that now, yeah. So I think it's very important that we keep emphasizing these sex bias problems as much as we can. And yeah, this is a good opportunity to thank you guys for what you're doing with this women's health research cluster, and it's been great to follow your processes and seeing all you do with blogs, and you know, everything is available. I'm always sending some new talk to my sisters and they're loving it, so it's really great, and thank you so much for doing it. And now we have to do similar things in Europe, and we have to keep pushing it, because it's still not common knowledge at all. It really isn't. So that's very important, I think. And then I think it's definitely becoming very critical that we extend collaborations, actually, because now we have lots of people working on exactly the same data. And you see it more and more, you see that similar papers come out, they're published within the same year, so they haven't even seen each other's work before everything is published, and nobody's cited each other, and it's often a little bit sad. But that's not strange at all, because people are working, on the same data. People have similar good ideas, across different labs. And I think this is sort of starting to become the norm a little bit. And I think we have to think about how to deal with it. We have to think about an updated literature search before we publish things, before it really goes to the latest stage of publication, which is something I can't say, I haven't really done that. When I'm busy with some revision, I just want something out, right? I'm not ready to go back to do another Google Scholar search, but I think it's becoming really critical that we do it. And then maybe you have to sometimes be a bit transparent and just reconsider the novelty claims, let's say, in your papers, if something else has come out that is very similar. And it's not necessarily a problem, I don't think. I just think we need to be aware that this is happening, and transparent and open for different angles to the same problem. And I also think that journal editors need to be on board as well and realize that, nothing is novel anymore almost. So just give us a break. And so another way of, helping with these kinds of things is obviously to organize bigger working groups together, working together across labs, and bigger initiatives basically for women's health research. There are already things ongoing, and I'm very positive to this. I think one challenge is that many of us don't have permanent positions. We don't know what we're going to be in the next years, right? So we are a bit reluctant, I think, often to start big things that we don't know if we can follow up. But I'm very hopeful that we'll just keep discussing this and coming up with good solutions. I'm hopeful. Okay, so just to summarize then, I hope that I've given you a general overview of some opportunities and some challenges as well when it comes to big data. I also hope that I have inspired you to look into the UK Biobank if you've not already used it. And I hope that the examples from our research were interesting and understandable, and that you get inspired to maybe look into these big data sets yourself if you're not doing it. And then lastly, I hope that I've made it clear that I really believe in this field, and I think we should move the field forward together. I think that is more and more key, and I'm very excited about that. And I think it's there's a lot going on. It's a very, very good place to be now, I think, because of how much movement it is and all these new things and all the new data, etc. So it's all in all very exciting, I would say. So then I will say thank you for listening, and thank you to, of course, the UK Biobank and the Swiss National Science Foundation, our great key collaborators across different institutions and countries. And with that, I am very happy to answer any questions or hear any of your ideas or comments.
Well, thank you very much. I already have a whole bunch of questions in the chat, so that's fantastic. I was going to just write. Feel free to put your question in the chat, but there have been a number of them. And thank you for those of you putting your little icons up there of the clapping, because, in this virtual context, it's always difficult. And I did forget to say, many thanks to you for giving a talk at 9 p.m. I was telling Anne-Marie earlier that I had to give one at 9 p.m. and everybody's like half asleep, but you did a fantastic job, and I really want to ask you my questions, of which I wrote down a whole bunch, but I should really give over to other people who have asked questions. Look at how many questions you have in here. Actually, and Amanda has the sort of the same question that I'm going to take as some prerogative. You can also unmute yourself if you want. Amanda is one of my trainees, so that's why I thought I could take prerogative and change it up. But she and I think I know the answer to this one. So how did you decide, like in your machine learning, I think this is what and Amanda, you can always correct me, machine learning, doing the brain age gap, how did you decide what's a normal aging? Number one is her question, because how do you decide whether they don't have any disease or any abnormalities? And number two is, Is there a difference in brain aging and parameters between males and females?
Very good questions. Excellent questions altogether. I would say that for the first one, yeah, it makes a lot of sense to think about it that way. But actually, because of these massive amounts of data, it makes very little difference. So it's quite interesting because we've thought about this many times. Goodness me, we must have some preclinical cases in here. What are we actually modeling? And although we cannot know for sure, we can't know how everyone is. So we only basically know that we can exclude people with diagnosis, right? But even when we include all of them, it actually doesn't make much difference. And I think it's because the mean is driving it so strongly. So you have most people who are just normal, basically, right? Or they represent the distribution, which is, yeah, more or less the average. And they drive this whole model setup then. So even if you add someone who is pretty far away, they will not actually influence it that much. But it would be very important, though, if you had smaller data sets and you, let's say you trained everybody on a small group of Alzheimer's patients, for example, and then if you test this model on another group of Alzheimer's patients, they would just look normal, like it wouldn't be any deviation. So in principle, you're absolutely right. Like I definitely understand what you're thinking, and it's true. But for these huge data sets where we have basically a lot of, yeah, I would say a population, a representative population in that sense, then it actually doesn't make that much of a difference.
And the male versus female?
Yeah, so what was that question? Can you remind me if there is differences?
You do, yeah. So if you do machine learning, because we already know there are a number of sex differences in the brain that show up all over the place, and there's not, and one brain is not inferior to the other, let's just put it out there. But are there sex differences in brain aging, and would that affect your model? So if you train it on male brains only versus female brains only, does that change?
Yeah, so I think it depends a little, but I mean, I think we did this in that study from Sivanya that I showed you, and we looked at it using the like sex-specific models where we only used males, only used females, and it looks very similar, right? So in the UK Biobank, we don't, yeah, we don't see huge differences really. But there are some other studies where they see more of a difference in brain age between males and females in general, right, at some time points. So I don't think I really have a good answer other than we have run it as a, you know, as a check, and we don't see a big difference. But I still think it makes sense to do it, right? It makes sense, especially if you're looking at sex-specific factors. I think it makes sense to compare females to females, right? Yeah,
exactly. And yeah, so the aging trajectory might not be different. They might start out at a different spot, but their aging trajectory doesn't look super different, maybe. So a question from I'm so sorry, I'm gonna get your name wrong. Josian. And it's a good question. Have you looked at adopted children only versus biological children? So if brain aging is reduced with parity, does it matter if they're adopted or not?
No, we have not. But this question, very often it comes up, and it's a very good question again. Yeah, it's definitely something we should do, or someone should do. And I think it could give us, you know, some indication of where, you know, what kind of mechanisms are we looking at here. But unfortunately, no, we haven't looked at it.
Is that variable in the UK Biobank? Do you know?
I don't know, actually, because I think one time in a talk I said no, and then I wonder if maybe I was corrected afterwards. I'm not sure. I'm sorry.
No.
I will look it up.
Emily asks, per slide 13, but I don't remember what it is, was age at childbirth and the associations of brain age investigated? So would a later brain age at birth, would that negate the positive effect on brain age because of other factors associated with aging? So is it, you know, more taxing on maybe on a person's body if they're older and they get pregnant?
Yeah, so that was actually quite tricky. Again, a very, very good question. We did look at age at first childbirth, because that's I think we have age at first and last. I think we've looked at both, but the issue is that it's quite correlated with number of childbirths, actually,
Right.
which makes sense, I guess. Yeah, it would have to be. So then we're struggling a bit with putting it into the same model because statistically it doesn't really work. But I think in the end we actually didn't see any similar result when we looked at only age at first or last birth.
And I guess another way to look at that might be, if you look at singleton births or two, and then you could look at that a little, maybe, sort of cap it, restrict it to those.
Yeah, I think one of the issues with these, I mean, these are all great questions. I think we often struggle a bit with very highly correlated variables, and we're not sure what to do about it. We want to look at the specific effects of something, but we can't really control for something that is very high correlated with it. Right. So it's not so easy always.
No, it isn't. Julia Sasha has the next question, which is slide 17. Julia, if you want to, you can ask a question out loud, or I can do it.
Sure. Well, first of all, thank you for a great presentation. So lucky to finally meet you, Anne-Marie. I was wondering regarding brain age, right? How functionally relevant it is, and if you had a chance to look at any measures in the UK Biobank to relate to your data, particularly for the sex-specific differences and throughout the menopause and with your adipose measures.
Yeah, so very nice to meet you too, finally.
My two favorite people are meeting. So great.
Yeah, that's great. Yeah, so what kind of variables were you thinking about then?
Probably cognitive.
In terms of cognitive performance.
Yeah, so we've looked at that as well, but we haven't actually been so good at including this, I must say. So yeah, we have some studies where we look at it, and it shows a relationship with brain age, as you would expect, but not necessarily super strong always, which is also maybe expected too. But we have not yet included this in the mix of adipose tissue measures or, body MRI.
Right.
Yeah, we haven't. Actually, it would be nice to have a follow-up chat about that, because I often struggle a bit with how to fit in cognition in the mix.
Yeah.
So maybe we can talk about that.
You probably know the LIFE study, right, in Leipzig, and it cannot compare in terms of numbers with the UK Biobank. It's 2,000 subjects, but we had a subsample of menopausal women where we actually find the relationship with cognitive measures, and very happy to talk to you more about that.
I want to be a fly on the wall in that conversation.
That would be great.
Thanks. You, Julia. Yeah, so much to say, but it's not about me. Claire Hill says, wonderful talk. Thank you. I am just wondering if anyone has done a genome-wide association study for your brain age data to look at significant genetic variants. Also a very good question. And then I want to see it by sex.
Yeah. Yes, it's also a very good question. I think there are quite a lot of genome-wide association studies, but we have been fairly restricted to things such as common variation when it comes to number of childbirths, for example. So there we used it to check, could this just be, some genetic variation? It's probably not completely random who has how many kids, etc. So I think we have been using it more for basically to rule out genetics, that's not what we're looking at. But for the brain age data in general, though, there are some studies, and it is heritable. So that's, I guess, was that a good answer to the question? Yeah, is this the Claire that I think it is?
It is, yeah.
No. Why are you tripping me up like that? This is my sister-in-law.
Because she knows. That's hilarious. To check you up on. Yeah,
Okay, I'm definitely sending you that study later then, Claire. Thank you for the question.
All right, and Natalie Sim is saying, thank you for sharing this exciting work. I know it is exciting work, isn't it? For your work on menopause, do you tend to focus exclusively on people with natural menopause? Another good question. Or did you include account for surgical menopause?
Yes, another good question. We have focused mostly on natural menopause, but actually I don't think, correct me now, anyone who, Sivanya or someone, if I'm wrong, Louisa, but I don't think that our results change drastically when we include people with surgical menopause, actually. But that could again be because we have so much power, and usually, you know, if you take 200 people in and out, it doesn't necessarily make a big difference. But I think both, you know, both angles are important. And I think it's a very good question, and maybe something we should think about more as well in terms of, you know, how we design these studies and whether we can, we tend to go for often what we have most data on too.
Right.
That
would be the natural.
Yeah.
It's Natalie here, if you don't mind me jumping in.
Yeah, no, please.
Sorry, Dr. Goya. Just to provide a bit more context, it's interesting to hear that the results don't change. I was thinking even for the analysis of hormone replacement use, particularly for the people that have surgical menopause, we know they tend to have more vasomotor symptoms and maybe prescribed HRT more frequently. So just even thinking about how that connects into the complexity of any hormone replacement therapy analysis is really interesting.
Yeah.
Fantastic.
That makes total sense. And I think in that analysis that I showed, we excluded anyone who didn't have the natural menopause for this reason, because it's a bit more complex than with the, with
An
the
earlier
hormone
age too. But Natalie, it reminds me of Julie Nineteen did a great talk. It's on our YouTube video on the many menopauses, which kind of blew my mind because I only thought of two. I had no idea there were more than two. So I think all of, you know, I'm sure all of it matters. Now we're running out of time. You have so many questions. You're going to win for the, not that there's a real prize, but for the most questions in one of these seminars, which is great. I'm really, really thankful for it, but we won't get to all of them. But maybe what we can do is have maybe we can entice you to write a blog where you answer some of the rest of the questions. We might be able to do that. Okay, last question I think that we'll have time for is from Elena. My question is about the study on abdominal adipose tissue and brain age. You said that longer reproductive spans were associated with older brain age, maybe for a greater lifetime estrogen exposure. However, you also pointed out that an earlier age for menopause was also linked to older brain age. Aren't those opposites? How do you handle those results when you find them? You go away and come back 4 hours later. You answer the question.
And this is that's a super complicated study, and I think the findings are also very difficult to communicate, and I probably didn't do the best job in the world. But basically what we see is that these associations, as you say in your question as well, are basically, they're more prominent in women with a longer reproductive span. So you can think about it in a sense that they're not, it's just a different, how do I best explain? If you think about, if you try to think about bins of women with different years of reproductive span, you will just see that these associations are more positive in those who've had a longer reproductive span. So I don't think it's necessarily opposite, I'm not sure, but I think it just tells us that whichever sort of hormone exposure that has been going on before the menopause might play into this. But we're not, I mean, we can't, we are speculating on what can it be and how is it linked, and we don't really know, because it could also be something specifically about the women who had a shorter reproductive span, or it could be a more general thing, you know, we just really don't know. It's very tricky, but there is a preprint out on this, so maybe, you know, I can forward that to you and you can try to read there. We have some pages to try to explain ourselves. But it's a good question because it's hard to make sense of these findings for sure.
Yeah, I have a whole bunch of things came into my head when you were talking about that, but I think I'll have to save that for another moment because we are now right on time, and I don't want to keep Dr. Lange delaying any longer because it's now 10 p.m. her time. So once again, if could you join me? Thank you so much for all your questions and enthusiasm. It is really terrific work, and I hope we can have more talks about this in the future. So thank you so much again.
Thank you so much for having me. This has been great.
Yes,
really nice.
We need more time though, more time. Anyway,
more time.
We'll get more time. Thanks everyone. Thank you for joining us.
Thank you so much, Lisa.